The blueprint of life, DNA, as our genetic code. We're all different in many ways. We have different hair color, eye color, and height. These differences make each of us unique, but deep inside our cells we share something remarkable and fundamental. This is a molecule called DNA, which stands for deoxyribonucleic acid. DNA carries our genetic information, the instructions for life. It's like a blueprint that determines our traits, from our physical appearance to our biological functions. DNA stands for deoxyribonucleic acid, a long and complex molecule. It's a long, complex molecule that holds the code of life. DNA is found in the nucleus of every cell in our body, acting as the control center. It's made up of building blocks called nucleotides, which are the basic units of DNA. These nucleotides are arranged in a specific sequence, forming the genetic code. The sequence of nucleotides in DNA is what makes us unique, giving us our individual characteristics. This sequence determines our traits, which are passed down from our parents to us. It's passed down from our parents to us, ensuring the continuity of life. DNA is essential for life, guiding the growth and development of all living organisms. It controls everything from our development to our daily functions, influencing every aspect of our biology. Understanding DNA is crucial for understanding life itself, as it holds the key to many biological mysteries. It's the key to unlocking the secrets of our past, present, and future, providing insights into our evolution and potential.
In 1928, a scientist named Frederick Griffith made a groundbreaking discovery. He was studying bacteria that caused pneumonia. Griffith was working with two strains of the bacteria. One strain was harmless, while the other was deadly. Griffith injected mice with the harmless bacteria. As expected, the mice survived. He then injected other mice with the deadly bacteria. Tragically, these mice died. This was all as expected. Next, Griffith heated the deadly bacteria to kill them. He then injected these dead bacteria into mice. To his surprise, the mice lived. This suggested that heat-killed bacteria were not harmful. Here's where things got really interesting. Griffith mixed heat-killed deadly bacteria with live, harmless bacteria. He then injected this mixture into mice. Amazingly, the mice died. This was completely unexpected. Griffith examined the blood of the dead mice from his last experiment, meticulously noting every detail. To his surprise, he found live, deadly bacteria thriving in the samples. But how did this happen? The deadly bacteria had been killed by heat, supposedly rendering them harmless. Astonishingly, the harmless bacteria had somehow transformed into the deadly strain, defying previous understanding. Griffith concluded that something must have passed from the deadly bacteria to the live, harmless bacteria, enabling this transformation. He called this mysterious phenomenon transformation, a term that would become pivotal in genetic research. But he didn't know what this transforming principle was, leaving a significant question unanswered. Today, we know that DNA was the transforming principle in Griffith's experiment, a groundbreaking discovery. The heat-killed deadly bacteria released their DNA into the environment. This DNA was taken up by the live, harmless bacteria, integrating into their cellular structure. The harmless bacteria incorporated this foreign DNA into their own genome, altering their genetic makeup. This process of transformation showed that DNA carries genetic information, a revelation that changed biology. It can pass from one organism to another, demonstrating the fluidity of genetic material. Griffith's experiment paved the way for further research into the nature of the gene, laying the foundation for modern genetics.
In 1952, Alfred Hershey and Martha Chase conducted another landmark experiment. They were studying bacteriophages, which are a type of virus that specifically infects bacteria. Bacteriophages are fascinating because they are viruses that infect bacteria, making them a perfect subject for genetic research. Viruses are much simpler than bacteria, consisting of only a few components. They consist of a protein coat surrounding genetic material, which can be either DNA or RNA. Hershey and Chase wanted to determine what part of the virus, protein or DNA, carried the genetic information necessary for replication. They knew that viruses inject their genetic material into bacteria, hijacking the bacterial machinery. This reprograms the bacteria to produce more viruses. essentially turning the bacteria into virus factories. To track the protein and DNA, Hershey and Chase used radioactive isotopes, which allowed them to distinguish between the two components. They labeled the protein coat of some viruses with radioactive sulfur, which binds specifically to proteins. They labeled the DNA of other viruses with radioactive phosphorus, which binds specifically to DNA. They then allowed these labeled viruses to infect bacteria, observing the process closely. After infection, they separated the viruses from the bacteria using a blender, which helped to detach the viral protein coats from the bacterial cells. They then centrifuged the mixture to separate the heavier bacteria from the lighter viruses, allowing them to analyze which component entered the bacterial cells. Section 5. Radioactivity enters the scene, tracking the genetic material. Hershey and Chase used a Geiger counter to detect the radioactivity. They found that the radioactive sulfur from the protein coat remained outside the bacteria. This meant that the protein coat did not enter the bacteria. However, they found radioactive phosphorus from the DNA inside the bacteria. This indicated that the viral DNA had entered the bacteria. Hershey and Chase repeated their experiment many times. They got the same results each time. The Hershey-Chase experiment provided strong evidence that DNA is the genetic material. It confirmed that DNA, not protein, carries the genetic instructions for life.
Section 6. The results are in. DNA is the culprit. The Hershey-Chase experiment was a turning point in our understanding of genetics. It provided concrete evidence that DNA is the genetic material. This discovery had profound implications for biology and medicine. Scientists now knew what molecule carried the genetic code. This opened up a whole new world of research. Scientists could now study DNA to understand how genes work. The Hershey-Chase experiment also had a major impact on the study of viruses. It showed that viruses use DNA to hijack the machinery of cells. This knowledge has been crucial for developing antiviral drugs and vaccines. Section 7. The implications: a new era in genetics. The experiments of Griffith and Hershey-Chase revolutionized our understanding of heredity. They established DNA as the molecule of inheritance. This paved the way for countless discoveries in the field of genetics. Scientists could now focus on deciphering the structure of DNA. Understanding its structure would be crucial to understanding how DNA replicates and how it codes for proteins. The discovery of DNA as the genetic material also had profound implications for medicine. It opened up new avenues for understanding and treating genetic diseases. Section 8. Beyond Griffith and Hershey-Chase, DNA's structure and function. Following the groundbreaking experiments of Griffith and Hershey-Chase, scientists were eager to unravel the secrets of DNA's structure. In 1953, James Watson and Francis Crick achieved a monumental breakthrough. They proposed the double helix model of DNA. Watson and Crick's model revealed that DNA consists of two strands that wind around each other. These strands are held together by hydrogen bonds between specific base pairs. Adenine pairs with thymine, AT, and guanine pairs with cytosine, GC. The discovery of DNA's structure revolutionized our understanding of how genetic information is stored, replicated, and passed down through generations. It laid the foundation for modern molecular biology and genetics. Section 9. DNA technology today, from medicine to ancestry. Today, DNA technology has become an integral part of our lives. It plays a crucial role in various fields, from medicine and forensics to agriculture and ancestry tracing. In medicine, DNA sequencing is used to diagnose genetic disorders, develop personalized therapies, and track the spread of infectious diseases. Forensic scientists use DNA profiling to identify criminals and exonerate the innocent. DNA technology has revolutionized agriculture, enabling the development of genetically modified crops with improved yields, pest resistance, and nutritional content. Ancestry DNA testing has become increasingly popular, allowing individuals to trace their roots and connect with distant relatives. Section 10. The future of DNA. Unlocking even more secrets. As we delve deeper into the world of DNA, we continue to unlock its secrets and explore its vast potential. The future of DNA research holds immense promise for advancements in medicine, biotechnology, and our understanding of life itself. Scientists are developing innovative gene editing techniques such as CRISPR-Cas9, which allow for precise modifications to DNA sequences. This technology has the potential to cure genetic diseases, enhance crop yields, and even resurrect extinct species. DNA computing, which utilizes DNA molecules to perform computations, is an emerging field with the potential to revolutionize data storage and processing. As we continue to explore the frontiers of DNA science, we can expect even more groundbreaking discoveries that will shape the future of humanity.